This video is about the origin of blood cell types and how to identify them. This chart shows the lineages and how they all come from hematopoietic stem cells. And then basically you have the myeloid lineage and then the lymphoid lineage. And I'm going to go into detail about all of these different types of cells. First, I'll start with the hematopoietic stem cells. All blood cells come from hematopoietic stem cells, which are found in the bone marrow. They're pretty rare. They stay dormant until the onset of infection. When you have infection, you need more blood cells to help fight it, so the stem cells are much more prolific under stress. They can become more hematopoietic stem cells, myeloid progenitors, or lymphoid progenitors. I'll start with the myeloid lineage. And common myeloid progenitors can become precursors for megakaryocytes, erythrocytes, mast cells, and granulocytes, which include the basophils, neutrophils, eosinophils, and monocytes. Megakaryocytes are really large cells that stay in the bone marrow and create platelets. Platelets are small cells or cell fragments that circulate in your blood and have to do with the formation of blood clots. They do not have nucleus. That's not to be confused with this picture, which is the megakaryocyte itself, not the platelets. The next type is erythrocytes, which is another name for red blood cells. Red blood cells have hemoglobin, which delivers oxygen to the cells in the tissue. And they don't have nucleuses because the progenitors leave the nucleus in the bone marrow before traveling to the peripheral tissue where they become erythrocytes. They're identifiable because of their shape. They are this unique disc shape which helps them be able to carry oxygen. Also, erythrocytes are able to release signals that induce immune activity when that part of the body is under stress. The granulocytes is a broad category of white blood cells. They all have multi-lobed nucleus, and they all have granules that carry proteins. They're used to attack pathogens. Some of these proteins attack pathogens directly. Some regulate other white blood cells, including the B and T cells, and some have effects on infected tissue, like they can heal it. Myoblasts are the precursor to granulocytes, which include basophil, neutrophils, eosinophils, and monocytes. You can tell up here all of them have the lobe nucleuses, and they all have granules. It's worth noting that mast cells also have granules, but they're not always categorized as granulocytes. Mast cells are released from the bone marrow as undifferentiated cells, which means they mature after they leave the blood. They're found in skin and connective tissue of various organs. Here's a picture of a stained mast cell, and then here's a diagram that shows its nucleus and the granules. The next one is neutrophils, which account for the majority of white blood cells, up to 50 to 70%. They have a lifespan of a few days, and they're first responders to an infection. So they are the biggest part of the primary response. Leukocytosis indicates an infection. That is when your white blood cell count is up, which is one of the things that a doctor looks for when you're sick. So after being released from the bone marrow, they migrate to the peripheral blood where they circulate for 7 to 10 hours and then migrate into the tissue. They're called to the site of infection by chemical signals or inflammatory molecules, which is generated by innate cells that have found a pathogen. Once they're in the tissue, they engulf bacteria through phagocytosis. Then they secrete proteins with antimicrobial effects and the potential to heal tissue. They're the main contributor to pus, which is where they end their life, which is one of the reasons you find pus around infected parts of your body. This diagram shows their multi-lobe nucleus and the granules, and they also have phagosomes, which is part of how they engulf the bacteria through phagocytosis. The next one is basophils, which are non-phagocytic, and they're identifiable because their granules have basophilic proteins. They're less common, but they have a big effect, so you don't need as many of them. They release the contents of the granules in response to binding of circulating antibodies, which is how all granules work. The biggest thing in their granules is a histamine, which causes allergy symptoms. This is easy to remember for me because I know that allergy medicine is called antihistamines. And in parts of the world where worms and parasites were more common, they have a huge role in defense against the worms. Eosinophils is similar to basophils, except they are motile and they are phagocytic, but their phagocytic role is less important than that of neutrophils. Their most important role is the defense against parasites, and we know this because they are most commonly found around worms whose membranes have been damaged by the proteins released from the eosinophil granules. They also have the ability to secrete cytokines, which regulate B and T lymphocytes and influences the adaptive immunity response. They are also contributors to allergy and asthma symptoms. The next kind of granulocyte is monocytes, which account for 5 to 10% of white blood cells. They migrate into tissue and then differentiate further. There are two basic types. There are inflammatory monocytes, which enter the tissue quickly in response to infection, and patrolling monocytes, which crawl along blood vessels and provide a reservoir for tissue-resident monocytes in the absence of infection. When they differentiate, they either become macrophages or dendritic cells. Macrophages migrate into tissue in response to infection, and some of them stay there and regulate repair and regeneration of those tissues. And some examples of these are osteoclast in bones, microglial in the central nervous system, and alveolar macrophages in the lungs. All of these just repair their respective tissue. Some of them are antigen-presenting cells that activate T lymphocytes. Once they're activated, the inflammatory macrophages are more effective. Macrophages and T helper cells facilitate each other's activation during the immune response, so they have to work together. Macrophages can express receptors for certain antibodies, and if an antigen is coded with the right antibody, the complex of this antigen and antibody bind to the antibody receptors on the macrophage more readily than antigen alone. This just means that phagocytosis is easier to accomplish. In this case, the antibody is an osponin, which is a molecule that marks the antigen for recognition for the immune cells. The other thing that monocytes can become is dendritic cells. They're characterized by their long membranous extensions that extend and retract to increase surface area for browsing lymphocytes. When they're outside of the lymph nodes, the immature dendritic cells monitor the body for signs of infection and capture antigens. And they process these antigens and then migrate to the lymph nodes where they present the antigen to naive T cells, which initiates the adaptive immune response. One interesting thing about dendritic cells is that they can come from the myeloid or lymphoid lineages. So there's two different ways that a dendritic cell can become a dendritic cell. Dendritic cells can carry out phagocytosis, receptor-mediated endocytosis, and pinocytosis. So they just pick up antigens any way that they possibly can and then take them to the lymph nodes. When they switch from being able to capture the antigens to the point where they present them, they lose the ability to perform phagocytosis, but their ability to activate T cells is heightened. Because once they pick up the antigen, they don't have to pick up anymore. They just need to take it. They just need to get the T cells there as fast as possible. So now we're to the lymphoid lineage, which that's where the common lymphoid progenitors become natural T cells or lymphocytes, which include T and B cells. Natural killer cells are the part of the innate immune system. They don't express antigen-specific receptors. They have two types of receptors, one that interacts with MH1 on healthy cells, and the other interacts with cell stress proteins found on tumor cells or other infected cells. They are distinguished by the expression of NK1.1 as surface marker and the presence of cytotoxic granules. Their sole purpose is to kill infected cells and cancer cells using granzymes and perforin. Perforin works by destroying or making holes in the infected cell's cell membrane. Here's a diagram of a natural killer cell, and the green circles represent the granzymes and perforin. They only kill cells that don't have MHCs, which means they kill anything that doesn't have a self-antigen on it. So they don't kill the host cells. And this is without any prior exposure. They just kill it on their first encounter. The lymphocytes are two different kinds, the B lymphocytes and the T lymphocytes. B lymphocytes get their name because they're made in the bursa in birds or bone marrow in humans. They have T cell receptors on their surface. They have B cell receptors on their surface, and they can recognize soluble or particulate antigens. They have the ability to become plasma cells, which are effector cells that lose expression of surface immunoglobulin and become specialized for secretion of antibodies. So these plasma cells are just antibody factories. They don't divide, and they live for 1 or 2 weeks. T cells mature in the thymus. They have T cell receptors on their surface, and they can only recognize processed pieces of antigens like peptides and are bound to MHC molecules. There's T helper cells and there's T cytotoxic cells. There's a few different types of T helper cells. Th1, Th2, and Th17 are a few. I'm going to make another video about these, so I'm not going to go too much detail here. Okay, here's some diagrams. Lymphocytes look basically the same. Under a microscope, the best way to identify them is by the receptors that they present. B cells do look a little different, and then plasma cells look very different. They're larger because they're basically just a factory, and the B cell has these different kind of receptors too. Okay, and I don't care if Dr. Blair uses this video in the future.